order to improve their coaptation ability and therefore minimize the mitral regurgitation incidence. So this adaptation ability is really fascinating, and one of the main players of this adaptation is the endothelial to mesenchymal transition. So this is maybe a little bit Chinese, but it's not too complicated. It just means that the endothelial cells get activated. And the activation of endothelial cells is something positive at the beginning, but when it's excess, when it's an excessive EMT, an excessive activation of the endothelial cells, we end up by having an excess matrix production and an increased stiffness of the mitral valves. This compromises the leaflet ability to bend and therefore their coaptation. So just as a little reminder, the mitral valve is constituted by endothelial cells, as you can see here. And in the interstitium, you have a lot of extracellular matrix, a lot of elastin fibers, collagen fibers. And in this interstitium, we also have some valvular interstitial cells. Of course, you have other cells composing the valve, but mainly these two players are the most important ones. So why is it so interesting to look into this EMT or endothelial cells or anything like that? It's because it has some clinical relevance. So if we manage to direct these adaptive mechanisms to just have an increase in the area of the valve and avoid these good mechanisms to go to a deleterious one and avoid the fibrosis, this might be a good way to limit the ischemic mitral regurgitation occurrence. And then there is also another aspect. There is this mitral regurgitation recurrence after repairing it. And maybe one of the mechanisms is that there is an intrinsic leaflet alteration, which is not treated. And we are just overlooking the cellular mechanisms there. We are just looking at the physical and the mechanics. And we should maybe look a little bit deeper into that to try to avoid this mitral regurgitation recurrence. So what is endothelial to mesenchymal transition? As we said, is just an activation of the endothelial cells. They adopt another phenotype, a phenotype closer to fibrocytes. They lose their cohesion with their neighboring cells, and they have the ability to migrate and start secreting some pro-inflammatory components. So this EMT plays a major role in the cardiac development. It's a positive thing, but it also plays a negative role, and it plays a role in the atherosclerosis, in pulmonary hypertension, and in many other diseases. One of the key signaling pathways that are initiating the EMT and which are really well studied are the transforming growth factor beta, the TGF-beta. And in one of the already published studies, which was a very elegant work, they showed that TGF-beta was a key player in inducing the remodeling of the leaflets in an ischemic environment. That was really interesting to see that actually the TGF-beta was doing that through the EMT by provoking the activation of the endothelial cells. But in our case, we decided to look into Tenascin-C. If you look a little bit into the publications of our group, you see that you can call us a little bit the Tenascin-C group. We worked a lot with Tenascin-C. So this is an extracellular matrix glycoprotein. Usually it's not expressed in adult healthy hearts, but it can be expressed at the embryonic development stage. It is upregulated after an injury, and it helps into trying to repair injuries, but of course, it is upregulated post-MI and is associated with cardiac remodeling. There's also something...
Hamza, excuse-moi, c'est bien. Samhi, est-ce que tu peux reprendre cette plaque en France? Cette plaque.
C'est là.
Allez.
Tenascin-C.
Voilà. Reprends, parce que c'est une plaque intéressante. Résume-nous en français. Si tu peux, hein. Maintenant, si tu peux. Parce que je pense que c'est là, c'est une transition importante.
Non, bien sûr.
Tu continues.
Donc en fait, la Tenascin-C, c'est une glycoprotéine qui forme la matrice extracellulaire. Normalement, elle n'est pas exprimée au niveau cardiaque, au niveau adulte. Mais en général, elle est exprimée à l'âge embryonnaire et cela permet le développement embryonnaire du cœur. Et ce qui a été déjà démontré dans notre groupe, c'est qu'on a on a une expression excessive de la Tenascin-C après un infarctus du myocarde et
Mhm,
ceci est associé à un remodelage du VG.
mhm,
Il y a aussi quelque chose de particulièrement important qui est intéressant pour nous aujourd'hui, c'est que la Tenascin-C a été démontrée comme étant une glycoprotéine qui joue un rôle dans la physiopathologie du rétrécissement aortique ainsi que son évolution. Autre chose, ce qu'on a aussi remarqué, c'est que la transition épithélio-mésenchymale,
ils ont chi-
euh,
Euh, Non, non, c'est bien. Mais là, tu as fait une bonne mise au point. Là, c'est bien. C'est bien parce que tu peux continuer comme tu l'étais.
en anglais ça ira.
De temps en temps, je t'arrêterai pour nous faire une petite.
C'est
Vas-y.
pas évident de passer au français, hein.
Tu as rien perdu. Tu as rien perdu, Sophie. Tu as rien perdu. Vas-y.
So, what I've also the really interesting aspect is that the epithelial to mesenchymal transition, which is the big brother of the endothelial to mesenchymal transition, which has been extensively studied. The endothelial to mesenchymal transition is something new; it's something relatively new. But the epithelial to mesenchymal transition is relatively old and well described. Um, you see that Tenascin-C is involved in inducing epithelial to mesenchymal transition in the metastasis formation, for example. So our working hypothesis was since Tenascin-C could induce epithelial to mesenchymal transition, and since Tenascin-C was involved in the cardiac remodeling after myocardial infarction, then probably Tenascin-C could also play a role in the leaflet remodeling in ischemic motor regurgitation, and this through EMT, the endothelial to mesenchymal transition. In order to do so, in order to study these mechanisms, we first had to establish an animal model of ischemic motor regurgitation. And in order to do so, we chose to work on a closed chest animal model. So in designing an animal model, we had the choice between doing it surgically or doing it percutaneously, meaning not opening the chest. Closed chest animal models are a little bit more delicate to produce, but they present the advantage of having a lower mortality rate, less adhesions, and in our case, it was really important not to have that much inflammatory processes as we can observe in an open chest model because we were on the verge of studying the inflammation at the valvular level. So if we used an open chest and had inflammation going on, it would have been a big bias. So what we decided to do to have an ischemic MR was to target the posterior papillary muscle. And to do so, we decided to inject selectively ethanol. But the main problem was to identify the irrigating branches of this posterior papillary muscle. And it was a little bit challenging knowing that there is an anatomical variability. So which branch is actually going and irrigating the posterior papillary muscle? In order to do so, in order to map the vascularization of the papillary muscle, we used contrast echo. We were injecting some contrast medium into some of the marginal branches of the circ and doing the echo in the meantime. After managing to identify the irrigating branches, we were injecting pure ethanol over the wire balloon inflated, which was avoiding having the backflush of the ethanol and going to the other coronary arteries. And after that, we were checking if the branches were occluded. If not, we were repeating the procedure and adding either half a milliliter or 1 milliliter, depending on the branch. So we had two sets of experiments. The first one was in the pigs. To establish our model, we chose to do that in the pigs first, and we followed them up for a short period, only 6 weeks. We enrolled 8 female adult pigs And we had a control group with weight and age match animals. 7 survived, but 1 had to be euthanized because of cardiogenic shock at 2 weeks. In the sheep, it was a longer follow-up period for 6 months, and we also had again 7 animals, and they all survived the 6 months period. So this is one specimen, one explanted heart, and you can see that we had, so this is the interior, this is the posterior view, and you see that we had an infarct, an inferior infarct involving the papillary muscle. As you can see here in the cut, there is an infarct. So these were the echo results. And you see that there was a significant reduction in the ejection fraction and a significant increase in the end-diastolic diameter, reflecting an enlarged left ventricle. So one of the pitfalls of this model in the pigs was that the pigs grow really fast. They gain 800 grams to 1 kilo per week. So it was a little bit challenging. And we thought that maybe the enlargement of the left ventricle was due to the growth of the pig, but actually it was not. When we compared that to our control group, which were weight and age matched group, there was a significant difference between the left ventricle and diastolic diameter between the ischemic MR group and the control group. So for the evaluation of the ischemic MR, it was a little bit delicate. We had to use some old parameters. We had to use index jet area, the vena contracta, because the PISA, the views and the quality of the images were a little bit not that great. Yeah, it's a little bit easier to do that in humans. So you can see that there is a significant increase in the jet in the index jet area. Yes, so sorry, I'm just looking into the questions just for 1 second because there is something really interesting here. So does Tenascin-C represent a biomarker of myocardial remodeling in heart failure? So yes, you have some really large studies in patients where they show that Tenascin-C, the circulating Tenascin-C, was correlated to the heart failure, but also to the myocardial infarction. Also, it was also a prognosis marker actually. So yes, and there is also another really interesting fact. When you treat mitral stenosis, it was also a predictor. Somehow, the Tenascin-C, circulating Tenascin-C, was also a predictor of restenosis later in the after treating mitral stenosis. So yeah, it's really an interesting question.
Is it on the animals or?
It's in patients. These are patients. It's really large cohorts, yeah.
Ah, okay, very interesting.
So yes, it's a really interesting question, definitely. Yeah, so we had significant increase in the index jet area, significant increase in the vena contracta, as well as the tendon area, which was reflecting a restrictive mechanism. It was a little bit the same for the sheep group, where we had a decrease in ejection fraction, left ventricle, which was enlarged, a significant mitral regurgitation, tendon area. It was all the same. The problem with this animal model is that it's really tricky when you have arrhythmias. External defibrillation was really inefficient. In our pilot study, we were really losing a lot of animals because of that. So we had to be a bit more aggressive in preventing those. One other possibility would have been to maybe use some internal defibrillation, but cost effective, I'm not sure about that. Another problem was the follow-up echoes. In the pigs, we had to sedate them, but in the sheep, we were able to do it in conscious animals. Well, it's a sheep. As its name says it all, they're a little bit nicer. But the problem was maybe the comparability of the results beyond the species. But the good thing was that the final examination was done in both animals under general anesthesia. And as I said, our quantification method to evaluate the mitral regurgitation was not the best. I see already the echographers here not too happy, but it's a little bit tough to do that on animals. But maybe in the next project, we were thinking about using the ICE, which is the intracardiac echocardiography. So what we could do, just put an indwelling catheter and do our follow-up through this catheter. So every week or every 2 weeks, we could go there, use our eyes, have a better picture, have a better view, and a better evaluation. Yeah. Yeah. So that was the first part of our experiment. The second part was to look into the really interesting part now, the valvular remodeling and the Tenascin-C expression. We expanded the heart, we harvested the valves, and we looked into their thickness. So as you can see here, it was really interesting. You can see already here that the posterior leaflet thickness was definitely significantly higher compared to the controls already, but also to the anterior leaflet in the diseased animals. This was after 6 weeks, and you can see a little bit the numbers here. That was really nice and interesting. After 6 months, not only the posterior leaflet was thicker, but also the anterior leaflet was thicker compared to the control groups, which was a little bit interesting. We moved to the Tenascin-C expression. We did some immunohistochemistry. Yeah, histology was really great. It was also interesting to look at that. You can also look here. It's just an overview. So this is the atrial side of the valve, and this brownish color here means Tenascin-C. That was really great. We found that Tenascin-C expression was more present in the posterior leaflets compared to the controls, but also to the anterior. But it was not only present after 6 weeks, but also persistent after 6 months. And one really interesting aspect was that this expression of Tenascin-C was well correlated to the thickness of the valves. So maybe there was a relationship between these two. So maybe Tenascin-C was actually doing something, but let's see. So this EMT, this bad guy, this good guy who becomes a bad guy, the endothelial to mesenchymal transition, to be able to see if it was there, if there was this phenomenon happening at the leaflet level, we stained the cells, the leaflets, with alpha-SMA and CD31. So the endothelial cell, when it's quiet, when it's non-activated, it expresses CD31. And CD31 is a typical marker of endothelial cells. Alpha-SMA is not supposed to be expressed by an endothelial cell. It can be expressed by a mesenchymal cell, a fibroblast, a little bit, and also a fibroblast which activated. And so if you see a cell going through EMT, it starts by expressing CD31, it's non-activated, it expresses CD31 and then also alpha-SMA at the same time. And at some point, it even loses the CD31 expression and expresses only alpha-SMA. So this is a little bit the stages of the EMT. So what does it mean for us? It's not too complicated. It's really simple. In alpha-SMA, you can see this alpha-SMA here along the endothelial layer, which means that maybe the endothelial cells were expressing alpha-SMA, and as we said, they were on the verge of being activated. And yeah, you can see that there is also a lot of alpha-SMA in the interstitium, probably some activated fibroblasts, some other cells which were infiltrating the valves. It was really interesting to see that. But then you can also see that CD31, which was supposed to be only on the endothelial cells, which were supposed to be just at the borders, they were also expressed in the depth of the interstitium, as you can see also here. So this looked like almost the endothelial cells were plunging from their nicely seated place to the interstitium. So these results are suggesting a valvular EMT process. We also looked into TLR4. TLR4 is an inflammatory surrogate just to look if there was inflammation or not. And something really interesting for us was that we found it in the endothelial layer, which means that, yeah, the endothelial cells also express TLR4, which will be interesting a little bit later in the cellular experiments. And we saw that, yeah, we had a lot of TLR4 expression in the ischemic motor regurgitation group, mostly in the posterior leaflet. This was reflect in some inflammatory process. So of course, histology is great, but yeah, we said alpha-SMA was along the endothelial layer. Was it really the endothelial cells, or maybe it was just some superposition of some endothelial cells and some other cell, a macrophage, interstitial cell, something else, which is, yeah, maybe. Another problem was also not a problem, actually a good fact. TLR4 expression was also superposed with the CD68. So CD68 is another marker of a macrophage, and if TLR4 and CD68 are both expressed by the cell macrophage, it means that it's an M1 polarization. It's simple. M1 is it's not that simple, but let's simplify it. M1 is a pro-inflammatory, a bad guy, and M2 is supposed to be a little bit of a good guy, the guy who is going there and trying to repair things. But yeah, in our case, it was more of a pro-inflammatory macrophage. Then, of course, the animal experiments were really great, but we wanted to go a little bit further, and we went into the cellular experiments. So we first isolated some endothelial cells from the mitral valves, and these were a primary cell culture. A primary cell culture means that you harvest them directly from a living tissue, and you have a primary and very delicate culture. It's not like these immortalized cells, which are a little bit easier to handle, of course, but they don't show the same characteristics as the primary cells. So we tried different concentrations of Tenascin-C with different time points, 1 microgram, 1 day, 2 days, when we went to 5 micrograms, to see if Tenascin-C in vitro could induce EMT. And in order to do that, we used some genes, and we looked into their expressions using real-time qPCR. And we saw that there was a significant upregulation of alpha small, MMP2, collagen 1A, and SLUG. SLUG is just an EMT transcriptor factor. So all of these genes are contributing to the EMT. So these results were pointing towards a mediated EMT by Tenascin-C in the MVEX. It was nice. Yeah, Tenascin-C induces MVEX, but induces EMT in MVEX, but how? We already talked a little bit about TLR4, and we know that Tenascin-C is actually an endogenous TLR4 activator. So TLR4 is involved in sepsis, and its usual activator, the activator is LPS, lipopolysaccharide thingy, which is a little thing expressed by some bacteria. It comes, it gets presented to TLR4. It doesn't bind directly to the receptor. It gets presented to TLR4. Boom, you have a sepsis. You have a whole cascade. But there is also some endogenous triggers to this TLR4, some danger, the stamps, these danger molecules, and Tenascin-C is one of them. So we wanted to blockade TLR4 to see if it would prevent the EMT, and indeed it did. So we had a downregulation, a reduced expression of alpha-SMA, SLUG, and these two new players, which are inflammatory markers and are also considered as markers of EMT. So it was really interesting to see that Tenascin-C mediated EMT was using TLR4 actually to do its job. So these two, TLR4 and Tenascin-C, could represent a potential therapeutic target for the future. Tenascin-C antagonism, it's still a big topic. It's not that easy to achieve, but TLR4, it could be a little bit more achievable and more into our reach, because you already have some TLR4 inhibitors used for sepsis, and yeah, it could definitely be used in the future. We already talked about the TGF-beta. We said that it's a well-known initiator of EMT, and we just wanted to use that as a side little experiment, to use it as a positive control, to say these genes were upregulated after stimulating the cells with Tenascin-C, Yes, but yeah, we also induce the same thing with the TGF-beta, and we know that the TGF-beta is induced in EMT, so this means that actually we really induce EMT. But what was really surprising, we didn't expect that at all after we used the TLR4 inhibitor in these stimulated cells with TGF-beta, was that it reduced the EMT gene expression. And it's not known that the TGF-beta has anything to do with TLR4. It's definitely nothing out in the literature saying that the TGF-beta binds to TLR4 and produces an effect. So we thought that maybe there was Tenascin-C again going in the mix. And after stimulating our cells with TGF-beta, we noticed that there was an upregulation actually of Tenascin-C expression. So maybe Tenascin-C was in the middle of this TGF-beta mediated EMT. But of course, we need to do a little bit more experiments to understand more how this thing happens. Cell experiments, they're nice, but...
Hamza, excuse me. You have two questions. The chat, if you can see the chat.
Oh, okay.
At the chat, you have a question about Mehdi and German. Preserved ejection fraction heart failure, a big problem.
So did you specifically identify other forms of Tenascin-C that are inducing this reaction that would be quantified into circulating blood? Tenascin-C, as you said, is a big problem. That's why we don't really manage actually to have an effective and proper inhibitor of Tenascin-C because it has such variability of it. Maybe I can show you that again. I didn't want to go too much into detail, but this region determines how different Tenascin-C can be. It's really difficult to identify which Tenascin-C, and it's definitely a big topic in our lab currently. So, yeah. But if you want to identify Tenascin-C, so you just choose which primer in the gene code, which one you want to amplify. So if you are unlucky and you go here, you might miss it. So you better, when you are designing your primers to do your PCR, try to avoid this area. Or if you choose to produce your primer from this area, you need to know that you are going to get this and this one, this variation of Tenascin-C. Did I clarify it enough?
Interesting question about preserved ejection fraction. Is Tenascin-C involved in preserved ejection fraction?
Yes. So Tenascin-C, yeah. So it's not quite exactly clear, but in our lab we are having another model. It's a TAC model. It's a trans-aortic constriction. So we provoke a constriction at the aortic level, and we have a hypertrophy with a, as you can say, you have a heart failure with a preserved ejection fraction. And we notice that actually there is a Tenascin-C involvement in the remodeling of the ventricle after having this hypertrophy. So,
German, you can speak German. Tu peux intervenir German si tu veux. German. On va laisser Hamza se reposer.
So the question is EMT. Is it involved with the stiffness, the myocardial stiffness in HESPEF, preserved ejection, heart failure with preserved ejection fraction, the EMT?
So yes, so the EMT. So you have so actually the EMT in the HFF would be more into the microvascularization. So you have a little bit of an involvement of it because you have some remodeling of these little microvessels going into the left ventricle. And it has some studies showed that, but not in patients. We don't have like clear markers of EMT going on there, but I already saw some involvement in some mice studies. And it's not about the myocytes because it's EMT. I don't know if you got a little bit the difference because EMT is really about just the endothelial cells. So it's just involving the vessels. So the vessels, you have the endothelial cells coating the vessel. So this is the involvement of the EMT. But the myocytes, they are going through another process, not EMT. They are going through apoptosis. They are going through another process in this disease. Actually, is it clear,
Okay.
gentlemen?
Go on, Hamza.
Okay.
Go on.
So
Quite.
we can talk about it later if needed, no problem. So one of the limitations of cell experiments is that, of course, you are in vitro. It's not, you don't have the whole environment that a whole organism could provide. And one of the problems was also that we had to have an extra step to isolate our MVEX, which is the MAX. MAX is the magnetic sorting of the cells. We use a marker, and there is a little antibody which goes and fixes itself on the receptor of these endothelial cells. And this antibody, on the other end, will carry a bead, a magnetic bead, and this bead will be retained in a little tube. And this is how we manage to keep these endothelial cells. It is problematic because whenever there is something binding to a receptor of a cell, we don't know if this cell will get activated or not. So it's a bit of a dilemma. Can we say that we only have endothelial cells by just using their phenotype, by just looking at them under the microscope and say, oh, yeah, my colony is only endothelial cells? Or should we have this extra step with the risk of activating them? Another aspect was also that we should have maybe isolated our cells based on the side of the leaflet. But again, this would have involved an extra step that manipulating too much the cell, it's not very nice for our study because it can also activate it. We used only indirect signs. We only used RT-qPCR. A more elegant way of showing EMT would have been to use FACS. So FACS can show the different markers. So if you have the same cell expressing CD31 and alpha small, this means that it is an endothelial cell which is going through an EMT. But the problem is that since we are working with pigs, yeah, the antibodies are not that easy to find. It's easier to work with cells coming from the mice, with murine cells, but swine cells, we really need to establish this protocol and work a little bit with the antibodies. So, as a conclusion, we show here that an excess EMT can lead to valvular remodeling, as we can observe in ischemic MR. What's new in our study was that Tenascin-C was identified as a contributor to valvular remodeling, and it was contributing to this remodeling by inducing EMT. And one of the mechanisms was the TLR4 involvement. This means that we could maybe use them as therapeutic targets to prevent or eventually limit the ischemic motor regurgitation. So the clinical perspective of this work would be, of course, to try to direct and modulate the cellular process to guide them towards an increased leaflet area rather than having a thick and not that flexible leaflet, which of course goes to motor regurgitation ultimately. So future therapies options might be local delivery of a TLR4 inhibitor, which would limit the EMT and subsequently the valvular remodeling. Or the holy grail, the Tenascin-C antagonism, could also be interesting in this setting because we would not only be targeting the leaflets, but we would also be targeting the left ventricle. So we could be taking the ischemic motor regurgitation as a whole, targeting the myocardium, but also the valves. So if anybody has any question, please do not hesitate.
Thank you for this very great work and this very interesting talk, inspiring talk. Great job.
Thank you.
Thank you. So if there is any question, you can write your question on the chat or directly to our speaker.
So I already have a very nice question. What would be the cutoff level of Tenascin-C in the heart tissue, or the fluids, or the needed time, duration present, to talk about the pathological signature? What would make Tenascin-C as a good biomarker for diagnosis or prognosis to follow up specific
Excuse me, Hamza.
Mhm.
Mahdi, you can
Yeah.
intervient directement. Mahdi, coucou. Voilà,
Reprends le micro. Bonsoir, bonsoir à tous et à toutes. Oui, oui, on est anglais en français. Sorry, I'm asking if. Allô? Vous m'entendez là? I'm asking. Yeah, I know that Tenascin-C is already used as a biomarker in many cancer situations already. And breast, for example, breast cancer and so on. But in the heart, it's very interesting what you presented for us. So thank you. And I was asking about the threshold or the cutoff level for Tenascin-C in the tissue or the fluids, where you could state or suggest that the situation is signature of for diagnosis or for prognosis. I don't know, because you spoke about duration time of 6 months, over 6 months. So I desire to know if it's possible that you could have different levels on a different period of time. So on this time series, would it be possible to state if it's pathologic or no? That is my question.
It's very interesting indeed. So what we've done, we cannot. I don't think that we can use Tenascin-C as a marker of for ischemic motor regurgitation. There are too many confounders there, and I didn't see so far maybe some studies with that. Numbers I cannot give you, didn't remember them, but in theory Tenascin-C should not be present at all. So the circulating in the serum or in the plasma, Tenascin-C should not be there, as well as in the heart, we should not be able to detect it, or really in small amounts, but really in small amounts. So there, you can find it in the chordae tendinis, or in a very very small amount in the basal membrane, but nothing more.
Maybe in severe aortic stenosis, I think we could see the remodeling reflect. It could reflect the remodeling of extravalvular cardiac damage. So
yes, sure. So when you have a pathological situation, yes, it reappears. So in the aortic stenosis, so you find it in the circulating levels. I cannot give you threshold. I'm really sorry. I don't remember. It's in the publications. But they also found it in the tissues. Actually, it was really in the tissues. So, but to say in the ischemic motor regurgitation having some circulating Tenascin-C means that we have a remodeling at the mitral valve. It's a little bit difficult because you have heart failure. Heart failure will definitely increase the Tenascin-C. You have myocardial infarction. Myocardial infarction is also known to increase the Tenascin-C, circulating Tenascin-C levels. So to quickly jump on that, because that was also a question. We looked into the Tenascin-C circulating levels in our animals, but didn't bring us that much information. So we just put it on the side and left it for a while. Because, yeah, it's a little bit tough to say. There is a local phenomenon which is involving Tenascin-C on the valve and also the myocardium. So both of them are producing Tenascin-C. So probably they will have to, of course, to decide if this is a prognosis biomarker, they will have to do some specific studies with this subgroup of patients.
Yeah, surely. I understand. I think that I have seen on BioRVix recently an article about that. If I found it, I will share it here.
Yeah, that would be great.
Okay.
Thank you.
Thank
Mr.
you.
Sliman, Berani, you can ask your question if you want. Berani, Sliman, you have.
Hi, everyone.
Yeah.
Well, my question is simple. Does Tenascin-C have any practical use in the management of ischemic mitral regurgitation, or is it still purely at the experimental stage?
So we are called the Tenascin-C group, and we are an experimental group, so it's still an experimental. So you will not be able to say, I will base my management decision, how to treat my ischemic mitral regurgitation based on the Tenascin-C levels, because I know that this patient will complicate and this one will not. So no, we're a little bit far from there.
Okay. If there is other questions to our expert on mitral regurg in the pigs and sheep. Y a-t-il des questions? Même en français. Bon, Hamza, peux-tu nous faire un petit résumé, pas trop long, hein, pour les nuls en anglais? Parce qu'il y en a encore, malheureusement. Donc, un petit résumé, sans trop... take-home message, un genre de take-home message en français.
Alors, tout ça...
Yeah.
Ouais, German. Vas-y, German, si tu veux parler. German. Dalia.
Yes. Of
course. Ask your question. German. Coucou. Vas-y, German.
Alors, maintenant, on dit que
l'essence mitrale
ne sont pas disproportionnés par rapport au volume ventriculaire. C'est-à-dire qu'il y a une très importante par rapport au degré de remodelage. Et c'est ces IM-là qui répondent bien au traitement interventionnel qui est le mitral clip.
Reprends ta question, German. German.
Là qui sont fibrosants.
On n'a pas bien entendu. C'était coupé, German.
C'est la suivante. Alors, on parle cette nouvelle mitrale, oui. Je crois que j'ai une mauvaise connexion. Je vais écrire. Je vais écrire.
Ta tête est dans
une mauvaise connexion.
Ah, d'accord.
Je vais écrire.
Écris alors, parce que tout à l'heure, on t'entendait bien.
Voilà, je vais écrire.
Alors, on t'entend là. Parle, on t'entend.
C'est bon? Vous m'entendez?
Oui,
est-ce que vous m'entendez?
Vas-y.
Voilà. Oui. Alors, ma question, c'est la suivante. Aujourd'hui, on sait que dans l'insuffisance mitrale ischémique, il faut distinguer les insuffisances mitrales disproportionnées pour pouvoir les proposer au mitraclip. Ce sont des insuffisances mitrales qui ne sont pas expliquées par le degré d'élargissement du ventricule. Et donc, c'est celle-là qui mérite d'être traitée de façon interventionnelle. Alors, ma question pour Dr Hamza, est-ce que justement, la concentration de ces molécules-là, fibrosantes, c'est une sorte en fait, de métaplasie de la base mitrale qui peut expliquer le degré vraiment de fuite de ces fuites mitrales-là?
I would love to be able to answer this question, but it's a little bit tricky because we didn't do any study. We did a little bit. We had some samples from patients, like 5 samples. We find Tenascin-C great, but we probably need to do a more extensive study to say yes, Tenascin-C is involved. This is when you have it, it's unfavorable for mitral clip. It's one of these settings. I cannot, unfortunately, answer this question.
Thank you.
Tu es à l'avance, Germain. Tu es trop à l'avance.
Je vais trop vite.
Tu vas trop vite.
Bon, y a-t-il encore des questions? Vous pouvez profiter. Tenascin-C, ça court pas les rues, hein. Tu sais, pour moi, c'est nouveau tout ça. Hein. C'est nouveau pour nous. Vous avez la chance, avant que je lui donne la parole pour nous faire le take-home message, On va poser vos questions, s'il y a des questions. Moi, je pense que bon, tu as été donc Hamza, tu nous fais, si tu peux, bon, on va, on va, on va te remercier après, mais là, si tu peux, on termine par un take-home message, si tu veux. Qu'est-ce qu'il faut retenir de tout ça?
Donc, ce qu'il faut retenir de tout ce charabia, c'est très simple. Les cellules endothéliales au niveau de la valve mitrale, lorsqu'elles sont activées, elles peuvent avoir un effet positif. Mais lorsqu'elles sont trop activées, lorsque ça va trop loin, ça provoque une fibrose de la valve mitrale. L'un des provocateurs de cet excès d'activation est le TGF-bêta. Tout le monde le sait, tout le monde s'amuse à jouer avec le TGF-bêta. Nous, on est parti regarder la Tenascin-C. La Tenascin-C, on a trouvé que finalement, oui, elle avait un rôle dans la fibrose et le remodelage de la valve dans l'IM ischémique. Et quelque chose de plus intéressant, c'est qu'on est parti un peu plus loin avec nos petites expérimentations avec les cellules. Et on a trouvé que la Tenascin-C avait cet effet d'activation des cellules endothéliales en activant TLR4. C'est un récepteur. Puisqu'on a plutôt du mal à avoir un antagonisme et on a du mal à bloquer la Tenascin-C, parce que c'est un peu compliqué, elle a plusieurs formes. Vu qu'on a trouvé le TLR4 comme médiateur, on s'est dit qu'on pourrait, si on n'arrive pas à bloquer la Tenascin-C, on pourrait éventuellement bloquer le TLR4 pour éviter ce remodelage. En gros, c'était ça.
Très bien. Très intéressant. Et ben, on te remercie beaucoup, hein, Hamza, pour nous avoir consacré un peu de temps. J'espère que c'est pas la dernière fois, hein.